All right, hello everybody. I'm excited to talk today about a device we developed for controlled, non-invasive deep brain stimulation. So what I want to go through is just sort of a background on transcranial focused ultrasound, and then discuss a problem in the field and how our research has addressed it. So transcranial focused ultrasound is an emerging way to deliver energy non-invasively into deep areas of the brain in a spatially focused manner. So we can constructively sum ultrasound waves into areas about the size of a grain of rice. And this new way of delivering energy is really effective and has a lot of potential for many different diagnoses and therapies. So for one, we can do direct neuromodulation with transcranial focused ultrasound. So we can change the activity of deep brain regions. With longer stimulations, we can actually create durable changes to brain activity and start to rewire pathological circuits in the brain. We can combine ultrasound with ultrasound-gated drug delivery vehicles like nanoparticles to deliver drugs like propofol, ketamine, doxorubicin, and many others into spatially specific areas of the deep brain. And another exciting application of this, which I think has a lot of potential, is using this for blood-brain barrier opening. So we can inject gas-filled microbubbles into the bloodstream and excite these with ultrasound to actually transiently open the blood-brain barrier. So we create small gaps in the endothelial cells that line the vasculature in the brain. And through these gaps, we can deliver stem cells, large drugs, and genes. So there's a lot of great potential for this method of delivering energy into the brain to deliver really game-changing therapies. However, the clinical translation of this technology has been hampered by the presence of a barrier to the field. And that barrier is the human skull. So trying to deliver ultrasound into the brain, once it propagates through the skull, we really have no idea how much energy actually was attenuated by the skull and now is being delivered into the brain. So we just don't know the dose of ultrasound that we're delivering into a patient. So to highlight this problem and to show you specifically what I mean, I measured the pressure field from an ultrasound transducer in the lab, and I overlaid it here onto an anatomical MRI just for scale. And so on the left, what you're seeing is the pressure field from the ultrasound transducer as I measured it in water. And on the right, you're seeing the same, you're seeing the pressure field that I measured from the transducer after it's passed through an ex vivo human skull. And so two things are immediately obvious. One is that the pressure field is extremely blurred, or the focus spot is extremely blurred. So this leads to off-target effects and reduces safety. And secondly, the ultrasound is unpredictably attenuated. So now we just don't know, we have much less acoustic dose being delivered into the target. And importantly, it's an unpredictable amount of acoustic energy being delivered. So this is obviously a huge problem. This is as significant as a problem as not knowing the amount of drug you're giving to a patient or not knowing the amount of electrical stimulation you're delivering with deep brain stimulation. So our group has come up with a method to actually address this problem. And our approach to deliver a known dose of ultrasound energy into the skull, into the brain, relies on this hardware setup here where we have two ultrasound arrays on the left and right sides of the head. And so on the left here, you see a simulation of the pressure field of this device. On the right, you see the device actually mounted onto a subject inside an MRI. So to go through the method of actually how we correct for the attenuation of the skull, the steps include, we first start out with measuring the pressure at target, just in water from each element of the device. We then mount the device onto a subject, and the presence of the skull introduces an attenuation factor A and a speed up factor tau, which distorts the incoming beam of ultrasound. And so our job, the whole goal of this correction is just to accurately determine this attenuation factor and the speed up factor, and then correct for it. And the way we do that is by taking through transmit measurements. So a through transmit measurement, we just send a brief pulse of ultrasound from one element on one side of the array and record the propagation with all the receiving elements on the opposite sides of the array. And we do this in the scenario in water, and we do this through the actual subject. And from the difference in these received signals, we can actually solve a system of equations and determine precisely the attenuation and speed up that the skull introduces. So once we determine those distortion factors, we can just correct for them by increasing the amplitude of stimulation and delaying the transmission time to account for those two distortions. So we're essentially erasing the effects of the distortions and returning the delivered intensity inside the skull to the same scenario that was delivered just in water. So this is an experimental setup that we came up with to actually validate this correction. We just directly measure the pressure field from our transducer array with a hydrophone. And we do this just in free field, which is through water. And we do this inside ex vivo skulls and under each different correction method. And so what you're seeing on the right is the output, which is the measured pressure field. We also measured this pressure field at 3 different disease relevant targets. And we did this to test the steering ability, so the ability of our array to electronically focus into different areas of the brain and address different brain targets. So the results of this experiment showed that our correction method was very accurate. So at our central target, just to step through the experiment again, at the we first measured the pressure field and free field just through water to determine the intensity that we actually wanted to send into the brain. Then we performed a hydrophone correction. So this is the best possible correction, the ground truth that you could achieve. And then without correcting for the skull, we measured the actual intensity that would be delivered into the target. And so it's important to point out that this intensity is an order of magnitude below the intensity that we actually wanted to send in. And this is what's currently being done with transcranial focused ultrasound therapies in humans. And then after applying our correction, we recovered 95% of the intended pressure at focus. And it's important to point out as well that our correction across the 8 skulls was statistically equivalent to the hydrophone ground truth correction. We found that this correction method was robust across different targets. Again, in each of these targets, we found that the correction delivered a statistically equivalent correction to the best case scenario. So overall, this correction method really overcomes the key barrier to transcranial focused ultrasound and really will allow us to deliver a known dose of ultrasound into the brain so that we can start translating these therapies into humans. So to summarize again, the human skull reduced the intensity at focus to 10.4% of the intended value delivered in free field. Our correction method restored the intensity to 94.8% of what we were aiming to actually deliver into the brain targets. And overall, this correction is critical to actually deliver a safe and effective therapy, safe and effective version of all of these therapies that are emerging today. So with that, I want to thank my lab and my collaborators, and I'll be happy to take any questions.
I was just wondering how much variability
Yeah, there's tons. So we actually had um, that's sort of the reason why the correction needs to, you can't just ramp up the amplitude to account for, like, you can't just send in 10 times more energy than you would to account for the skull. It's because each skull varies so much that uh, by doing that, you'd often like overshoot with skulls that are thinner and undershoot with skulls that are thicker. In 8 samples, we had 2 with bone metastasis, which is apparently relatively common. This is just outgrowths of bone in the skull. So each skull is just very different, very complex.
I was wondering if you did any studies looking at differences in ultrasonic frequency, or so we didn't talk about frequency, but I know that can affect the transmission properties, and was wondering if that was a part of the effort for this application, which is very different, where you really want to focus and also, cause some kind of physical disruption, rather than doing something that you don't want to cause any disruption.
Yeah, that's a great question. Frequency is a huge component to delivering ultrasound through the skull, because higher the frequency, the more the ultrasound is attenuated by the bone. So we have to stay below around 1 megahertz to actually get any reasonable amount of energy into the brain. And what's interesting too is that with this correction method, the lower you go in frequency, the easier it is. There's less phase shifting. There's just less distortion. We're trying to balance that out with finding the right parameters that are good for neuromodulation. So just kind of conflicting evidence where you can see that neuromodulation is more effective at higher frequencies, but sometimes it's, or in some cases, it's been shown to be better at low frequencies. So we're trying to deliver, for now, the highest frequency we can while not being attenuated too much by the skull.
My question is, so right now you create this like really cool tool to kind of focus it inside the brain. What's kind of the next steps for this? Are you trying to improve this model, this tool, or are you trying to like move over into kind of more animal studies, trials like that?
We actually are moving right into humans with this. We have an IRB to apply this for neuromodulation in patients with treatment-resistant depression. So we're going to be doing some safety studies first, applying this device on them.
Hey, thanks. Great talk, Tom. I'm interested in the relative importance of the change in amplitude versus the change in phase. You mentioned it briefly talking about the frequency, but is it if you do away with the change in phase, just ignore it completely? What are the effects look like?
Yeah, that's a really good question as well. So the phase, if you don't do any phase correction, you'll get roughly like 80% of the intensity at focus. If you don't do any attenuation correction, that's when you get like 20% of the intensity at focus, and those two multiply. So attenuation is much more dramatic of a change, and the phase shifting you could really get around by going through thin parts of the skull, which is the left and right sides, as opposed to like front and back. So yeah, the attenuation is a much harder problem, and phase shifting can be gained around just by lowering the frequency.
So that's with respect to the amplitude. You didn't talk about like the tightness of the focus.
Oh, right.
Which fine, but does the phase have more of an impact on the?
Yeah, the phase almost completely determines the size of the focus spot.
Do you expect any additional factors that you need to implement into your model when you go into in vivo with all the extra tissues?
Yeah, that's really the strength of this method, is that we directly measure all tissues in the beam path. So hair, scalp, any bubbles, how well it's acoustically coupled to the patient. So it's a really a direct measurement of attenuation through all these things. So that's already included in the model. And the only other ways to correct for the skull are through CT imaging. And that's sort of the weakness of that methodology, is that they just can't account for these other obstacles.
How much does the brain move with just like regular, like this kind of blood? Does that ever happen?
I'm really not sure, honestly, about how much the brain is moving. I doubt it'd be more, well, our targeting has an accuracy of like around 2 millimeters, which is sort of determined by the wavelength of the ultrasound that we send in. So, and the regions that we're actually targeting are around the size of like 5 millimeters. So as long as we can be sort of in that range, I think we're fine. And we're also doing, to actually target these certain areas, we do like sort of a functional registration, where we stimulate the areas and see if we get the effect that we want. If we don't, then we move to a different area.